So good afternoon, everyone. It is my honor to introduce today's CTSI distinguished speaker that all of us know very, very well, Dr. Jake Luces. Dr. Luces obtained his Ph.D. in biophysics from Oregon State University and did postdoctoral training in genetics at the Roswell Park Memorial Institute in Buffalo, New York. Thereafter, he was recruited to UCLA, where he is a professor of medicine, microbiology and molecular genetics, and human genetics. Dr. Lucis's laboratory uses the combination of genetics, molecular biology, and informatics to investigate pathways underlying common cardiovascular and metabolic disorders. His group has a plethora of areas of active research that I cannot summarize here, but will say that these include the interactions between lipid metabolism and inflammation in cardiovascular disease and the metabolic syndrome, as well as the identification of genes and pathways leading to heart failure. The Lusis lab has taken advantage of high throughput technologies to dissect complex traits. In particular, his group has shown that the integration of high throughput methods with natural genetic variation can be used to model biological pathways and uncover regulatory mechanisms, an approach known as systems genetics. During the past decade, the Lusis lab has developed a unique mapping method in mice that is analogous to genome-wide association studies in humans. By exploiting natural genetic variation in a well-characterized population of more than 100 inbred strains of mice, termed the Hybrid Mouse Diversity Panel, or HMDP, this experimental approach provides high-resolution mapping and permits the scrutiny of gene-by-environment interactions, which are difficult to address in humans. This has led to the identification of novel targets and a deeper understanding of disease pathways. Dr. Luces has been honored by multiple prestigious awards, including the Duff Award, as well as the Distinguished Scientist Award from the American Heart Association and the Anichkov Prize from the European Atherosclerosis Society. One quick housekeeping item, please use the Q&A feature to type questions, and I will gladly ask them to Dr. Luces after his talk. And now please join me in welcoming Dr. Luces for a CTSI lecture entitled Sex and Tissue-Specific Regulation of Mitochondria, Role in Heart Failure with Preserved Ejection Fraction. Jake.
Thank you. Thank you very much for that very nice introduction, Renee. I want to thank the organizers, people that invited me, and also the people that are attending this seminar. So today I will discuss mitochondria and cardiovascular traits. But I'd like to begin with maybe 15 or so minute discussion of our approaches and interests to sort of give you background to our studies. I began, well, I did my postdoc, as Renee mentioned, in mouse genetics with a guy named Ken Pagan at Roswell Park. And when I came to UCLA, shortly after I came, maybe a year or so later, I thought I had a good idea. And this was to look at common complex forms of cardiovascular disease and metabolic disease using the tools of mouse genetics. So these common forms have a significant genetic component, but they're not Mendelian. They're due to the interactions of multiple genes. And it's been a long time since I did my postdoc and since I came to UCLA, but this is still pretty much the focus of our lab. We do human studies as well. We try to extend our mouse research into humans where possible, but I would say that this is still the focus of our lab. And that's what I'm going to tell you about, how we use these tools to look at, in this case, mitochondria and cardiovascular disease. So the experimental design, I guess the main experimental design that we used for a couple of decades is shown here. We would take strains of mice that differ in traits of interest, let's say atherosclerosis. C3H are very resistant, C7 black 6 are very susceptible. We would cross them to obtain F1 hybrids, and then we'd intercross them to get a segregating population of mice. right? A segregating population. And what we would typically find is a continuum, let's say from very small lesions to very large lesions. And this would be the case because the trait is due to many different genes that segregate independently. So each of these mice inherit different regions from C3H and C57 black 6 that affect atherosclerosis. And we were able to, for example, map the trait by examining genetic markers in all of these strains to examine which regions of the genome came from C3H or C57 black 6. Sort of a major advance for us in the entire field, of course, was about 20 years ago when methods were developed for high throughput omics type technologies, such as transcriptomics, proteomics, metabolomics, and so on. And we have used these to integrate with the clinical traits. So for example, in an F2 cross, we could examine transcript levels in a relevant tissue in each of the mice, each of the segregating mice. Right? This would allow us to map loci that control transcripts. We could ask if the transcripts map to the same locus as the clinical trait. We could do correlation. We could do network modeling and so on with traits like this. So we use that extensively starting about 2000. One way to think about this is that we're looking at the flow of information from DNA to transcript to protein, metabolite, and so on. About 10 years ago, we switched our sort of experimental design or a resource that we're using. Instead of looking at different inbred strains of mice, we switched to sort of looking at a population type based approach, association mapping. So we set up a set of about 100 common, well-characterized inbred strains of mice. Holy smokes, I'm sorry about that. Let me turn off my phone here. About 100 inbred strains of mice, and that had been mostly sequenced, high density genotypes, and so on. And this allowed us to have several advantages. One of these is that it allows high resolution mapping, much better resolution than you get with linkage analysis. It also allowed replication. All the members of an inbred strain are genetically identical. So we could look at as many members of a particular strain as we like, get better data. We could look at genetic interactions, as Renee mentioned. For example, we could look at a particular genetic background, an inbred strain, under a variety of conditions. And then, of course, systems genetics, as Renee mentioned. This is very convenient because if we carry out omics technology on these 100 strains, the data is kind of cumulative. We could use it for other studies. And using this approach, we were able to map genes and study variations. So for example, one of the first variations we looked at was response to a high-fat, high-sucrose diet. Right? So we looked at males, females, 6 to 8 mice each. Here in the black are shown the levels of body fat on the chow diet, and in the red are after feeding for 8 weeks on the high-fat, high-sucrose diet. And as you can see, there's a lot of variation, and we're able to map this using association. It's analogous to genome-wide association in humans. And so, for example, here, if we're looking at the change in body fat in response to the diet, we see 1, 2, 3, 4, 5, and there are 2 loci here, 6, 7 loci, right? And the loci tend to be fairly narrow, sometimes a megabase or so, but we can identify genes. We've identified some of these genes. And so the method works, right? And we can apply systems genetics very nicely. But if you compare this GWAS to a human GWAS, So this is for coronary artery disease. There's no comparison. So by looking at tens of thousands or hundreds of thousands of individuals in human studies, it's possible to identify many, many more loci. There's much greater power in human genetics than in our mouse genetics. So this is for atherosclerosis. I think they're all together here, about 100 loci. By comparison, here we are seeing loci, but we're only seeing the strongest loci. We're not seeing all of the loci. It's a matter of power. And as you know, human studies now, GWAS studies have been done for literally hundreds of traits. There are thousands and thousands of loci. Many of the loci have, no obvious genes, and so it's a great data set for future studies, right? Investigating loci and so on. It's not always easy, but I would say that this has definitely been a success. So one thing I thought I would talk about today, that something that might occur to some of you, is given the success of human studies, why study the genetics of complex traits in mice? Why don't we just go straight to humans, right? And I think that there are several reasons. I'm all in favor of human studies, but there are several reasons, I think, to study mice. And a couple obvious, right? In humans, experimental studies and access to tissues are very limited, right? You can't do any kind of invasive experiments for ethical reasons. You can't access tissues such as blood or skin or maybe adipose tissue or other tissues from people that have died, such as in GTEx, but access is pretty limited, right? And so let me show you an example of how the mouse can inform us with this example. It's a study that I very much like. It's a collaborative study that was done together with Henry Sukoff, who was then at USC, now he's at University of South Carolina, and Michaela Patterson, a graduate of UCLA, who was a postdoc with him. Henry was very interested in heart development and in heart failure. If an individual has a heart attack, the cardiomyocytes, the cells there, will die, and that will become a scar. Mature adult cardiomyocytes cannot regenerate and replicate, right, in the adult. And this is a big problem because this can very often lead to remodeling and heart failure. It's a huge problem. So cardiomyocytes during fetal life in the embryo can proliferate and regenerate, but they lose this ability shortly after birth. And Henry noted that just around the time they lose this ability, they become multinucleate. And he hypothesized that maybe mononuclear cardiomyocytes may have some ability to replicate and regenerate. And so he decided to test this using the hybrid mouse diversity panel, the set of 100 strains. And so he looked for the frequency of mononuclear cardiomyocytes. And it was very interesting that some strains of mice had 2% mononuclear cardiomyocytes. Other strains had about 15%. A very significant variation, right? So this, again, is not something that one could do in humans. Also, he was able to do experiments directly based on this, right? Again, something we couldn't do in humans. So what he did was he injured mice with a small number of mononuclear cardiomyocytes or a large number. He found that they all lost ejection fraction. Ejection fraction is the fraction of blood that is expelled from the left ventricle with each beat of the heart. So this went down from 70% to 30%, right? But then the strains with a lot of mononuclear cardiomyocytes started to recover, right? Consistent with his idea. And as I mentioned, you can also map. He was very lucky because he had a very small locus with a small number of genes, and he identified a kinase here that turned out to be part of the regulation of mononuclear cardiomyocytes. He looked at strains, knockouts, heterozygotes, and wild type, and clearly the knockouts had more more mononuclear cardiomyocytes, right? So he could look at traits that wouldn't be possible in humans and do experiments. Another reason, second reason why it's worth studying mice, genetics of complex traits in mice, is that you have much better ability to do omics level analysis, right? Frequently, human studies are limited to, as I mentioned, blood or skin and so on. And you can't collect that under, ideal conditions, right? I mean, individuals have different environments and so on. So you can't do this in mice. And since we're talking about heart failure today, let me just show you one sort of unpublished study that we've done. So we looked at hearts across the HMDP using proteomics. Ben Parker in Melbourne, Australia, did very sophisticated proteomics across the strains, the soda chow diet, and Marcus and Krista and Karthik in our lab were involved in these studies. And what we were able to identify we were interested in mitochondria and heart failure, the topic of this seminar. And what we found was that there are several major loci that contribute to mitochondria in the heart, and they're tissue specific, we think. One locus here on chromosome 13 affects the assembly of complex 1. All of these are complex 1 proteins. Another locus here, CoQ7, affects coenzyme Q metabolism. All of these are related to coenzyme Q metabolism. Another affects the translation of mitochondrial RNAs. There are specialized processes for that. And there's another good candidate. And interestingly, each of these variations are associated with heart rate. Decreased mitochondrial function increases heart rate, increased hypertrophy. So we could relate it to functions. Now we know that this is part of the explanation for the variation in mitochondria and heart. We can look in humans, we can look in other mouse studies. A third aspect, important reason for using mice, I believe, is that now some of the human geneticists may jump on me for this, but I believe this is the case. Gene by environment and gene by gene interactions tend to be lost in human genetic studies. I really haven't seen much in human genetics that is very enlightening in terms of gene by environment. You can do sort of statistical analysis and so on, but I think a lot of this gets lost. There's a very nice review by Sackton and Hartle in Cell 4 years ago that discusses why human studies are poorly powered to look at gene by environment interactions and gene by gene interactions. And yet in mice, we see them everywhere where we look, right? So for example, if we look at body fat change in response to a high fat diet, some strains don't gain any weight. Some strains gain a ton of weight, right? And we see this for all kinds of traits, right? So we think that gene by environment and gene by gene interactions are going to be very important, and that they may be best studied perhaps in mice. We also see lots of gene by gene interactions. If you have a knockout and you put it on different genetic backgrounds, more often than not, the phenotype will differ. And then there's a final thing here at Reason. Certain important traits just can't be studied in humans. So for example, this is where we've studied food consumption, right? Why do some strains eat more food than other strains? This is males, females, you can see pretty much the same loci. There are other traits. For example, there's a student in the lab now that's studying exercise. If you put in a running wheel, some strains will run 10 kilometers, some won't run at all. So what's the difference? So these are interesting traits that could be studied in mice that you really can't examine in humans. Okay, so with this very long introduction, let me turn now to the topic that I want to discuss today. And that's mitochondria and cardiovascular traits. I think you probably all know about mitochondria. They have two membranes. They are the powerhouse of the cell. They have electron transport, ATP generation, the TCA cycle, and so on. Mitochondria have their own DNA. It's about 16,500 nucleotides in both mice and humans. The DNA encodes about 37 genes, and there are about another 1,100 or perhaps more genes that are encoded by the nucleus that are mitochondrial. Right? So we have studied mitochondria in a variety of tissues in the hybrid mouse diversity panel. And we found something very interesting. There's a lot of variation. And part of that I've explained to you. There are just genetic variations, like in complex 1 or coenzyme Q1 metabolism, that affect mitochondrial copy number, apparently, as well as function. But we also see very significant sex differences. So here is adipose. You can see that males or females have more mitochondria copy number than males. It's just the reverse in liver, where males have more. Males also have more in heart. So there's this interesting difference that, it hasn't been seen because you can't look in humans, right? And we don't know if it happens in humans. I would guess it does. And interestingly, we've related each of these variations in mitochondria and the sex difference in mitochondria to clinically important traits in adipose, obesity and insulin resistance, in liver, fatty liver disease, and in heart failure. So today, I'm not going to talk about all of these. I'm going to talk about heart failure and mitochondria. But I want to just give you one slide each from these to give you a little bit of background. Okay, let's look at adipose. So here is adipose. Let's see. This is mitochondrial copy number here, and this is fat mass. So this is in mice, the hybrid mouse diversity panel. And what you can see here, as I showed you before, females are the ones that tend to have very high levels of mitochondria, and you'll notice they have very low fat mass. In fact, in females, there's a very strong correlation between mitochondrial copy number and fat mass. The mitochondria, based on R squared, explain about 30-35% of the total variation in body fat. A huge amount. You also see this in males, I'm sorry. You see this in males. They have less mitochondria, but you also see that as well. So this is a very interesting correlation. Is it causal? Does being obese affect mitochondria in adipose or what? So one way to address this is sort of like Mendelian randomization. You can ask, are there variations that affect mitochondria, and do they in turn affect body fat? And yes, there are. Karthik identified a locus on chromosome 17 that affects all kinds of mitochondrial genes. It also affects mitochondrial copy number, and indeed, that same locus affects fat mass. Same locus. So we think that this is probably, at least in part, causal. It may have to do with the phenomenon called browning. So that's sort of energy wastage due to uncoupling of mitochondria that occurs for sure in females. Okay, that's adipose. Let's look at liver. So in liver, with Shaw Yang, we did sort of network modeling. We had expression, we had fatty liver phenotypes. And what we found is that a lot of the genes, the key drivers that she calls them, are associated with mitochondrial functions. This is, for example, a protein that affects cristae formation. Here's pyruvate kinase that's involved in glycolysis. So these very much go with mitochondria, mitochondria go with fatty liver. And there's also a sex difference, So what we found is that some of these genes, like pyruvate kinase, are sex regulated. And indeed, we've actually validated all of these genes experimentally. Indeed, in males, but not in females, the levels of pyruvate kinase go with triglyceride levels. Okay, so this takes me to the topic of my talk. I hope I have enough time here. And this is, now we're looking at the genetics of heart failure with preserved ejection fraction. Right? This is a project taken on about 2 years ago by Yang Cao, who's a postdoc in the lab, senior postdoc, a really, really outstanding student. She's really been the one doing the thinking here as well as the work. And so that's what I'm going to talk about her studies for the rest of the time. I don't know if you've heard about heart failure with preserved ejection fraction, but it's probably the most important unmet need in all of cardiovascular disease, right? It's very common, and we know almost nothing about it. I'm on a study section, not a study section, a committee at NIH to develop RFAs for heart failure, but it's a real big deal. So we decided to take it on, or Yang did. And let me just tell you a little bit about heart failure. It's, of course, the inability of the heart to pump a sufficient amount of blood. Affects about 7 million people in the US, very common. People don't live very long with heart failure. Common forms are highly complex and heterogeneous with important environmental factors. And so it's really been very difficult to study. It's only been recognized not that long ago. So there are two major types of heart failure. One of these is heart failure with reduced ejection fraction. The heart muscle does not contract effectively, so you don't expel as much blood from the left ventricle as you should. The other is preserved ejection fraction, heart failure with preserved ejection fraction. Now the heart muscle contracts, but it doesn't fill normally, right? From the blood comes in from the atrium into the ventricle, and it doesn't come in as well as it should. It doesn't relax. That's at least one of the characteristics of heart failure with preserved ejection fraction. Okay, now, let's say a little bit about it. It's about 50% of all heart failure, maybe even more. Very common, huge. No therapies. You know, there are several clinical trials that have been done, and some therapies work a little bit on certain forms and so on, but really, I mean, the only therapies are exercise and diet, really, at this point. Thus far, there are hypotheses, fibrosis, inflammation, impaired fatty acid oxidation, and so on, but they're all hypotheses. They're all hypotheses. And one very interesting difference that we have focused on, that Yang has chosen to focus on, is a sex difference. There's a striking sex difference. Females develop heart failure with preserved ejection fraction about twice as frequently as men, right? And the females also tend to have more severe disease. Very striking thing. And so we've been interested in this. Now, we approached this problem using data that we had generated in a collaboration with Yibin Wang's lab, UCLA. We had two students that were involved in it, Christoph Rao, who's now an assistant professor at North Carolina, Jessica Wang, who's an assistant professor at UCLA. Jessica is an MD, Ph.D. And so what we did in this study was we infused isoproterenol. It's a beta adrenergic, it's an agonist, and it stimulates the heart to beat more rapidly, right? Over time, the heart remodels, hypertrophies, and results in some strains in a heart failure-like phenotype. And so we studied this. We did echoes and so on. Jessica and Christoph, and members of Yibin's lab. And we found, you know, for example, the left ventricle increased in size, hypertrophy. This is after isoproterenol, and this is before. And you could look at lots of variations, and the data is still being studied. Christoph in North Carolina, Yibin, we're all still studying these data. But what we decided to do is to look at a different aspect of this, and that is genetic variation related to relaxation, to diastolic function, when the heart relaxes. Now you can study this using what's called pulse wave Doppler, PWD. With pulse wave Doppler, you can measure blood flow velocity within a small area at a specific depth in the myocardial tissue. Let's say right here. And when you do that, what you find is that there are two waves that you see. One occurs when the ventricle relaxes. The other occurs when the atria contracts, right? So you see these two peaks. The ratio of those peaks is a measure of relaxation. So for example, the A peak will be lower if the ventricle is stiff and it can't contract very well. There's another measure that I'm going to discuss. It's called E to E prime, and that measures the velocity of the cardiac wall, right? It's just another measure. So both of these are measures of relaxation. The higher the measure, the poorer the diastolic function or the relaxation. So we see variation in this as well in this HMDP treated with isoproterenol. Here it is after treatment, and here it is before. You see variation in both. You can map this, just as I showed you. You can map things. So there are two major peaks, one on chromosome 4, one on chromosome 10. In this case, it's been unfortunate because both peaks are rather broad and have a lot of genes. So this is really a pretty tough situation if there aren't any genes that look like good candidates, right? So we have an idea that was actually suggested to us by Arjun Deb in cardiology to look at this using a technique that was developed by Mike Tatel's lab, Mike, director of the Cancer Center. It's called live cell interferometry with phase shift of light. And we've used that before to measure contraction or relaxation of cardiomyocytes in the study here. And so what we've thought about doing is to do this, look at tissue culture cells, knock down genes using siRNA, and look for changes in relaxation. It's been technically challenging, but that's still something that we would like to do. It's an ongoing project. The other thing that we came away from this study that made us think about certain mitochondria is that if you look at the pathways associated with E to A ratio in this HMDP, mitochondria are the strongest, right? Mitochondria are the strongest, TCA cycle, mitochondrial inner membrane, that really came out. And so that started us thinking about mitochondria and heart failure. And so as I told you, males and females have a difference in sex difference. Females tend to be susceptible, much more susceptible to HFpEF than males. And we thought, well, could mitochondria be involved in this, right? Are females more susceptible because they have lower mitochondria? Now, I have to say, we don't even know if this occurs in humans, but it does occur in mice. Why do they have lower mitochondria? Well, it's probably mainly hormones. So if we take a male on a chow diet or a high-fat sort of metabolic syndrome type diet, and we gonadectomize it, mitochondria go down. If we add testosterone, mitochondria go back up on both of these diets. It's also regulated by estrogen. If we take a female and we gonadectomize it, mitochondria go up. Add estrogen, they go back down. Okay? So it looks like it's controlled by sex hormones. It'll be interesting to think about this in terms of menopause and so on. And males, of course, lose testosterone as they get older. So anyway, what Yang then did was develop a mouse model in our lab, a mouse model that had been reported about 2 years ago by the Hill lab in Dallas, UT Southwestern in Dallas. And it's kind of a two-hit model. You make the mice fat by putting them on a high-fat diet. And you also inhibit nitric oxide synthase using a drug called L-name. This causes high blood pressure, hypertrophy, perhaps aortic stiffness, fibrosis. This causes body weight gain, glucose intolerance, and so on. And what you get out of this is a model of HFpEF that looks like HFpEF, at least in some respects. Okay. And this Shaw started off, or Yang started off by looking at originally C57 black 6 mice. And on this diet with L-name and high-fat diet, they become insulin resistant, they become fat, of course. The E to A ratio increases. So diastolic function is impaired, and E to E prime also increases. Running also increases, the ability to run, but ejection fraction remains the same. Okay, so then because of this sex difference, we decided to focus on sex differences, and we looked at males and females, and you can see females have more E to A ratio, more E to E prime ratio. Left ventricular function is, however, normal. They also have more, lung weight is just a water accumulation, right? What happens in heart failure? Females are worse than males, and the two-hit model worsens the heart failure. This is also a measure of heart failure. And finally, and related to the other stuff, the mitochondria, the diet decreases mitochondrial levels in both males and females. And of course, females already start off lower in terms of mitochondria. Now, this is just looking at mitochondrial DNA. The key thing is mitochondrial function. And this is, I have to thank Laurent Vernis in Karen Rui's lab for these studies. He's an expert in mitochondrial function. And in these studies, he used an instrument called the seahorse. And I won't go through it because I'm running a little short on time. But if you look at oxygen consumption rate, this is coupled state coupling to ATP production. Complex 1 coupling, this is where you add ADP and phosphate, looking at ATP production. You can see it's clearly lower in females than in males. And it's reduced by the two-step model of HFpEF. The same thing, these are all uncoupled. You can look at electron flow in complex 1, 2, 5, look at maximal. You use various substrates for this. Here we used palmityl carnitine. Here we use succinate and so on. But you can see, big difference. Females have on top of the mitochondrial mass, a big decrease in mitochondrial function. And if you go and adectomize the mice, this is just looking at males. In males, of course, testosterone increases mitochondria. And testosterone also, if you go and adectomize them, is involved in mitochondrial function. Function is reduced. Okay? So to look at this using genetics, now this is just something we've started, and there's a lot to do. But what Yang did was look at 30 strains of male and female mice on this high-fat plus L-name diet. She fed them for 5 weeks, looked at echocardiogram, electrocardiography. We euthanized them. Females, again, as we saw before, and with black 6, have higher E to A and E to E prime ratios. So they have poorer diastolic function. There's a lot of variation here. You might think, is it genetic? Yes, it is genetic. It's highly heritable. E to A ratio, E to E prime ratio, fractional shortening, elongation fraction, they're all heritable. This is genetics we're looking at, including genetics because of sex differences. You can look at all kinds of traits. So there are lots of things you can look at. For example, we find diastolic function correlates very strongly with insulin resistance. So it's clearly there's a strong metabolic connection. Right? Strong. This is something that we need to follow up on. And you can look at other things like this is totally out of the blue. So if we look at in our data set mononuclear cardiomyocytes, fraction of mononuclear cardiomyocytes. That also correlates with the E velocity, right? Why would that be? We have no idea. I have no idea. But you get all these clues that you can follow up on. You can generate hypotheses using these data. And what we've done is in the studies that Yang has done so far is focus on sex differences. So if you look at, this is just black 6 here, control, the high fat, the HFpEF diet, males, and here's females. And you can see here that these are certain genes involved in certain processes. We see that females show a decrease in certain genes, or an increase in certain genes on the HFpEF diet that males don't show. There's more inflammation, more cell cycle. This is a very attractive area to look at, right? Could very well be involved. We've been sort of focusing on fatty acid metabolism and mitochondrial function. And you can see that that goes down here in the females, but not so much in the males. And one gene that I'm going to talk about is this ACSL6, right? So I'll end up looking at that. Now, what Yang did, there are lots of ways to look at these big data sets. She decided to do kind of an overlap type thing. We looked at a human study with males and females of heart failure, about 1,300 individuals. The data is available. We looked for sex differences, differential expression in heart failure. We looked at a study in the HMDP, sort of a metabolic syndrome type study, mitochondrial gene sex differences. And then we looked at this 2-hit HFpEF model, differentially expressed sex differences. And we come up with certain genes. This is the overlap here. You can look at these things lots of ways, but you come up with genes. And we came up with one that we've been studying, and I'm going to end by telling you about this. ACSL6, it's an acyl-CoA synthase long chain fatty member 6. It acylates fatty acids, long chain fatty acids, using CoA and the fatty acid and ATP. And these then can go into lipid metabolism or lipid biosynthesis or beta oxidation. What you see is the enzyme is lower in females. The messenger RNA decreases on the HFpEF diet in females and males, but females start off much lower. Gonadectomy affects this, but most convincing for us was this. So we of course have looked at messenger RNA levels in all of these inbred strains, and so we can map loci the control transcript levels. Okay. These can be regulated locally or cis or transregulation. So ACSL6 has a cis, what we call expression quantitative trait locus. Near the gene are variations that go with expression. AA genotype is low expression, GG genotype is high expression. This is this particular SNP that we see that's right next to ACSL6. So it's being driven by, perhaps promoter variation, enhancer variation. That same SNP associates with E to A ratio. Okay? Higher expression of ACSL6 results in improved diastolic function. Right? So it's an interesting candidate. So we've studied this by overexpressing it initially in AAV9 and adenovirus. We use the troponin T promoter, which is specific for heart. We express it or GFP. And what we find is, yes, E to A ratio is improved if we overexpress it in heart. And E to E prime is expressed. The mice can run longer, right? It's kind of interesting. There's somewhat of a decrease in heart weight to tibial length. There's no change in ejection fraction. So it looks like it's a gene that influences, it's one gene whose variation affects this relaxation, one aspect of heart failure with preserved ejection fraction. So this is where we stand right now. Just to summarize the studies Yang has done, in genetic studies of heart failure in mice, diastolic function was most strongly associated with reduced expression of mitochondrial genes. And in fact, mitochondrial copy number goes with diastolic function. In a mouse model of HFpEF, females have reduced mitochondrial function as well as impaired diastolic function as compared to males, suggesting that the mitochondria may explain the sex difference in the disorder. An integration of human and mouse data, hard data sets, primarily expression data, identified mitochondrial genes that are causally associated with this diastolic function. Yeah. So we're looking at a couple. The one I told you about is this ACSL6. So we think that then this suggests something that we can look at in humans, that females, because they may have lower mitochondrial function, or mitochondrial function may be impaired, mitochondrial mass being impaired, that may predispose them to this very important and very common disease. It's something we can now look at. Future studies, well, of course, we want to do the make knockouts and overexpression and so on to look at individual genes. But apart from that, there are certain things like, for example, how do mitochondrial genes and pathways affect diastolic function? How do they affect the relaxation of cardiomyocytes? We'd like to follow up on other pathways associated with HFpEF, such as injury, cell proliferation. We certainly want to examine the relevance of mouse findings to human HFpEF. There probably will be very large studies of HFpEF. There have been some. So are our data consistent with what's found, at least in certain forms of human HFpEF? And one of the things particularly that mouse studies are useful for is identifying biomarkers that may help distinguish different forms of HFpEF subtypes, for example. We've done this previously. In the isoproterenol study, we identified a heart protein whose expression was dramatically affected by heart failure. It's a heart protein and it's secreted. And we looked in the blood and we could see the same difference. And then we tested it in human heart failure subjects. And it turns out it's as good as any marker there is, marker for heart failure. This is with reduced ejection fraction. So we want to do the same sort of thing here. I think that would be a big advance. And I think I've mentioned most of the people that are directly involved in this. These kinds of studies, of course, are a team effort, are technicians. Zanang has done a lot of the work on this and so on. But it's a team effort. Yang has really sort of totally led the HFpEF studies that we've done. Former students have been involved in some of the earlier data and so on. We have lots of wonderful collaborators at UCLA. This is one of the great things about being at UCLA. It's a very friendly place, and there are so many terrific people that have expertise in all kinds of things, and it's great to collaborate with these people. And with that, I'll take any questions if there are some.
Jake, thank you so much. That was unbelievable overview of the power of the HMDP, and also very important work on HFpEF, which, as you mentioned, is really an unmet clinical need. So understanding the genetic architecture of this disease is very, very important. So we have a lot of questions as anticipated. So one question from Steven Cedarbaum is, can you please clarify what you mean by mitochondrial copy number? How do you count mitochondria? Do you mean mitochondrial DNA copy number?
Yeah, Steve, yes, exactly. It's mitochondrial DNA copy number. And we simply look at it we have primers for mitochondrial DNA and we have primers for nuclear DNA. And we do the PCR and we just look at the ratio. So it's the ratio of mitochondrial DNA to nuclear DNA. It's just a very convenient measure. It doesn't tell us function, but it seems like in most of the studies that we've done, the mitochondrial DNA copy number reflects function pretty well.
Great, thank you. We have another question by Sami Yanikian, who's asking whether there are plans to look at calcification in the heart with these models.
That's a great idea. That's a great idea. And we actually see calcification, particularly in these heart failure models. In the study with with Ibin's lab and looking at isoproterenol, we saw clear evidence of calcification. We identified one gene that's involved called ABCC6. And that's another really, really good candidate that we, or calcification is a really important trait. We just haven't looked at that yet. But definitely that's high on our priority list. Yeah.
So another question by Ari Srivatsan is, can the mitochondrial functions change with exercise after this two-hit model you showed of HFpEF?
That's a great question, Hari. Yeah, we haven't looked at that. There is a student in the lab and who is studying exercise, and he actually has looked at this. If mice exercise a lot, then there are changes in mitochondria. So I wouldn't be at all surprised. That's a really good idea. And again, as I mentioned, one of the, about the only thing you can do with HFpEF is exercise and diet. Yeah, that's a great point. That would be interesting.
So next we have a question from Marlene Tuma, who's asking, does mitochondrial copy number associate with increased mitochondrial biogenesis or turnover or both? And how would that apply to HFpEF?
It's a good question. Good question, Marlon. That's something we could look at. I might give a shout out to the new metabolomics theme and Orion Shirahai, who has really brought expertise in mitochondria to UCLA, and that's really what's been a driving force for us to get into this area. But I think that's something that could be looked at, and that's a really interesting question. Yeah.
So we have a question also from Lara Bidayan, who's asking what might be the mechanisms of the sex hormones enacting this phenotype that you were showing?
Oh, the sex, well, I mean, clearly the hormones are affecting mitochondrial copy number, which is, for me, it's unexpected. I mean, why would males have more mitochondria? Why would testosterone and estrogen regulate it? There are people that have looked at this. Mitochondria, they're regulated in lots of ways, genetically, but other ways of regulating it. And Andrea Hevener's lab here at UCLA in endocrinology has done quite a few studies there. And so we haven't looked at the mechanism for the regulation between sexes in heart or adipose, but that's an interesting point. That would be interesting as well. Yeah, one could perhaps, come up with drugs that affect mitochondrial copy number if that was really a beneficial thing.
Another question, Jake, is have you seen or looked for any correlation between the percent mononuclear cardiomyocytes you were showing from your collaboration with Henry and mitochondrial gene expression?
Yeah. What I showed you was just a correlation with mitochondrial copy number. But mitochondrial copy number, if you look, it's some the electron transport genes, in all of our studies very closely sort of reflect mitochondrial copy number. So, technically we haven't looked at that, but we've looked at lots of those relationships in other contexts. But, that's a good point, really. What genes change and so on. There may be some specificity there. So that's another good suggestion that we should.
Yeah, each of these questions, of course, is another year or two of experiments, but they're very pertinent questions, very interesting questions. So I think we have time for maybe a couple more questions, and actually the last one by Mite is the one I wanted to ask you to, so I'll leave that one for last. But we have a question from Madhuraka Pal, who's asking, since metformin increases mitochondrial biogenesis and is the most widely used therapeutic for diabetes, could it potentially be therapeutic for cardiovascular disease and maybe the models you showed?
What a good idea. What a good idea. Yeah. metformin is used for treatment of diabetes, and what a great idea. I hadn't thought about that. That's a really good idea. Thank you for that suggestion.
And the last question we'll take from Mete, and hi Mete, is actually the same one I wanted to ask you.
Is this Mete Civilek?
Yeah, our Mete.
Oh, wow. So he's on this. Mete, hi.
He's on, right.
Mete is an associate professor at University of Virginia, and he was a postdoc in our lab, and we miss him.
Yeah. So he's asking, are there genetic variants in mitochondrial genes that are associated with heart function?
There are, yeah, but they tend to be rarer variants. As I mentioned, there are 37 genes that are encoded by the mitochondrial DNA, including important members of electron transport chain. And yes, there are. I think that they tend to be more Mendelian. They're associated with a lot of traits, with all kinds of traits. But I think what we're looking at here is primarily nuclear encoded genes. The mitochondria obviously have to talk to the nucleus, and a lot of the regulation is nuclear. We don't have any evidence that what we're looking at is really mitochondrial. For historical reasons, a lot of the strains have very similar mitochondria. But that's a good question. That certainly could be involved.
Okay, Jake, I think that that wraps it up. So thank you so much for your presentation. It was excellent as usual. I've heard a lot of your presentations, and each time I learn something new. You do generate also a lot of data at a very rapid pace in your group. And also, I think what's even more important is, and I can speak about this firsthand, is and Mete and so many people who are now faculty and very successful, have their own careers, are really all the folks that you've trained, who have been successful scientists in leading and prominent institutions. And I think that stems from your approach to science. And I can speak about this firsthand, which is really, and I don't say this just, to say it, it's one of really genuine passion. Like you're a passionate scientist, and you've always been driven by just trying to understand things, dissect them, break them into smaller pieces to try and analyze those smaller pieces. And it's I certainly have learned a lot by interacting with you and from attending your seminars. And I just want to thank you for that, and thank you for your collaborative spirit. Thank you, really.
Wow, it's very nice of you to say that, Renee. Thank you very much.
Okay.
Thanks to everybody.
Thank you, everyone.